Hello, could somebody confirm you can hear me?
I can hear you.
Great. Why is that coming up? All right, before we begin, are there any questions? All right, I'm not hearing anything, so I'm assuming there's no questions. Let me state that I have done some grading. I don't think I mentioned that last Tuesday, but I did grade the last lab and the last lecture worksheet. Let me take a look at that. Yes, the last lab, lab 9, you may make corrections by 11:59 p.m. this Saturday. That's August 26th. I also graded, I think it was lab 4. Let me look here. Yes, I've graded lab 4 for the second time. So that has been graded now for both the first time and the second time. I'll try and work on lab 5 and 6 tonight. Any questions about the grading? Any questions about anything? All right, let me get out of the grade book and then I'll share my screen. How do I get out of here? There we go. Let me remind you that I am an instructor, so my job is to help teach you. If you need help on something, please let me know. The greatest joy that many instructors, including myself, get out of teaching is helping my students learn. All right. Am I sharing my screen? I don't think so. Come on.
No,
Yeah, I'm trying to get it shared now. There we go. All right, so today is August 24th, and we should be on chapter 13 and finish it today and start chapter 14. You'll note there is no lab today. I will be checking in for 15 minutes to answer questions, but after 15 minutes, I will log out unless, of course, there's long and lots of questions. You should be working on your unknown project. The unknown project is due when Wednesday, August 30th. The unknown notes page will close on the 29th. And if you ask for a test on the 29th, it'll have to be a test that I can get to you by one day. If the incubation time is longer than a day, then you will not get the results in time to turn in your project. So try and ask your tests before that day, especially if it's a test that takes 2 or 5 days. Any questions about any of that?
do
All
we
right.
have lecture on next Thursday?
No, there is no lecture on whatever that day is, the 31st.
Yes.
You have the comprehensive final on the 31st. Let me talk
Okay.
a little bit about the final. I'm surprised that isn't up here. 50% of the final questions will be on material since the last quiz. So the last quiz covered chapter 8 and lab 9. So 50% of the questions will be on chapter 13, chapter 14, and then the unknown project. The other 50% of the questions will be on chapters 1 all the way to chapter 8. Generally, each chapter will have a question or two, not counting chapter 13 and 14. They'll have a fair number of questions. One exception is chapter 1. Chapter 1 goes over a lot of introductory topics that we then touch on later in the term. And then chapter 1 also goes through the history of microbiology. And as you can probably tell, I really like the history of microbiology. So I do tend to ask a few more questions from chapter 1. Any questions about any of that? Not sure what the chat's about. If the question was on what percentage of the exam questions will be since the last quiz, it's 50% of the exam. Any question about any of that? The best way to study for the exam is to go over the chapter objectives. So each lesson has an objective, so go over those. If nothing else, do that. Obviously, do that and then go over your notes if you have the time. All right, any questions about what we're doing today? I'm going to try. I don't think I'm going to succeed. Come on. I'm having trouble with my mouse. I'm going to try and finish as much of chapter 14 today as I can. I won't be able to finish it, but I don't want to have a lot of material on Tuesday to cover chapter 14 because you have the exam on Thursday. I'm sorry about that. The summer really is truncated. There is no finals week in the summer. All right, I have that we discussed COVID-19 last time. I just read something. There's a new strain of COVID-19, which is highly mutated from the existing strains. And it has been recently introduced to the United States. And they're watching it because they're afraid that this COVID-19 will spread and become a majority of the cases of COVID-19. And they think because it's so highly mutated that if you're vaccinated or you've had COVID-19 before, that you will not be protected against this strain because it's going to be so different from the strains that are currently going around or the strains that the vaccine has been made to. And they're not sure because obviously this is a new strain, but this is what they're thinking about COVID-19. And I don't remember the name of this new strain of COVID-19. It's given initials like BA28.9 or something like that. All right, let's talk a little bit about viruses and cancer. Some human cancers appear to be caused by viruses, and this is about 10 to 15% of human cancers. Some examples, it's well known that human papillomavirus causes cervical cancer and skin cancer. And it's well known that hepatitis B virus and hepatitis C virus cause liver cancers. A recent study by Tang, which I guess that's not so recent anymore, but in 2013, he found that breast cancer and glioblastomas were not associated with viruses. So not all cancers are associated with viruses, but some of them are. Let me briefly mention that cancer is uncontrolled cellular growth where your own cells grow uncontrollably. There are two main ways that viruses can cause cancer. First, you can get a formation of a provirus, where the viral DNA integrates into a chromosome of the patient. So your chromosome. And that virus generally integrates fairly randomly. That will be a permanent genetic change in a cell. And when the DNA does that, it can turn on genes that should be turned off. It can turn off genes that should be turned on, and it can mutate genes. And you can see where the formation of a provirus could lead to cancer. The other way a virus can cause cancer occurs with an oncogenic virus. This is a specific virus that carries an oncogene. An oncogene is a mutated form of a normal cellular gene that's involved in cell growth. This mutated gene doesn't act normally, and it causes the cell to grow uncontrollably, like a cancer. And so the mutated oncogenes help transform normal cells into a cancerous cell. This oncogene probably was at one point a normal cellular gene involved in cellular growth, and it became mutated and then picked up by a virus. And then when the virus infects the cell, it causes this cell to grow uncontrollably, and that allows the virus to replicate. And so you can see why the virus picked up that gene. Any question about any of that? All right, those are the two ways that a virus can cause cancer. Integrating into the host DNA, mucking up the DNA, and then if the virus carries an oncogene, it will lead to the loss of cellular growth and regulation, normal cellular growth and regulation, and you'll get uncontrolled growth. And that we call an oncogene. I mentioned that in animal cells, they can be following the lytic life cycle, and they can also follow the budding life cycle. We can also have viruses which follow the latent life cycle. This is an animal virus, by the way. And we can have viruses that follow the persistent viral infection life cycle. Let me talk about first the persistent viral infection. The persistent viral infection life cycle of a virus is one that is very similar to a budding virus. The difference is the persistent viral infection is a naked virus, and the budding virus has an envelope. The budding virus buds out of a membrane of the cell, which is either the cell membrane or the nuclear membrane or the membrane of the endoplasmic reticulum. A persistent viral infection life cycle, when the virus replicates, it doesn't bud out of the cell because it's a naked virus. What it does is it leaves the cell by exocytosis, meaning the cell puts the virus, or several of them, in a, what do you call that? A vesicle. And then it moves the vesicle to the cell membrane and then dumps the contents of the vesicle outside the cell. And that's how a persistent viral infection replicates. Generally, a persistent viral infection, that life cycle, reproduces the virus at low numbers, similar to a budding virus that buds at low numbers. So usually the virus doesn't kill the cell, and it's not lysing the cell, so it's not killing the cell that way. The virus is released by exocytosis. It's called the persistent viral infection because usually the patient has this virus for a long period of time. And then they're shedding the virus at low numbers for long periods of time. Any question about a persistent viral infection life cycle? Another virus life cycle I want to talk about is the latent virus life cycle. The latent virus life cycle is mostly a virus that has the budding life cycle. And this budding virus stops the budding life cycle and then enters the latent life cycle. When the virus is in the latent life cycle, the virus remains asymptomatic in the host cell, and it can remain asymptomatic for a long period of time. When it's in the latent life cycle, the virus does not cause any disease, no signs or symptoms of disease. The virus is actually fairly inactive when it's in the latent life cycle. And then the virus can leave the latent life cycle and then enter the budding life cycle, causing signs and symptoms of disease. An example of that is a cold sore virus, herpes simplex virus type 1. When you have the cold sore, you are in the budding life cycle. But then the cold sore goes away, and that's because the virus goes into the latent life cycle. And then later on, you can have the virus leave the latent life cycle and enter the budding life cycle, and you'll have another cold sore. Any questions about that? An even better example of a latent virus is the chickenpox virus. Usually, chickenpox strikes a child, and the child gets signs and symptoms of chickenpox, and then the virus goes away. It doesn't totally go away, it becomes latent, and it stays in the ganglia of the child, and generally the virus stays latent for a long period of time. Doesn't have to, but it may stay latent, and usually it stays latent for decades. And then the virus leaves the latent life cycle when the child is now an elderly adult, and the virus comes out as the shingles. Every time the virus leaves the budding life cycle, after the first time, it will be shingles, meaning the first time you get this virus, it's the chickenpox. Every other time you get the virus, meaning when it comes out of the latent life cycle, it will cause the shingles. Any question about any of that? Usually there is a trigger for the virus to leave latency, meaning the latent life cycle, and then re-enter the budding life cycle. And the trigger is usually a stress. For herpes simplex virus type 1, that stress is usually the patient comes down with a cold, and then the patient gets a cold sore. It can be another stress, though. For shingles, you won't like this one. The stress is usually the patient becomes old, their immune system weakens, and then the virus leaves latency. But there can be other stresses. Any question about any of that? All right. A persistent viral infection is a disease process that occurs over a long period of time. Detectable infectious viruses may build up over a long period. Often, I shouldn't say usually, often the virus is never cleared by the host immune system. It may be fatal. AIDS used to be a persistent viral infection of HIV that would become fatal. Now we do have medication to keep the HIV patients alive for close to the patient's normal lifespan. The patient, assuming they're taking the medication and the medication works, you can keep the patient alive for close to their normal lifespan. They do have a little shorter lifespan. I don't remember if it's a couple of years or 5 years less, but it's fairly close to normal. And that's obviously in the first world country where you have access to the medication. But before, HIV was a persistent viral infection. Disease never went away. This is the old days before the medication. The virus was never cleared by the host immune system. The virus actually destroyed the host immune system, and then it was fatal. A patient would then get another infection and would die of that other infection. Another example is subacute sclerosing panencephalitis. It's actually caused by the measles virus. And the virus in this case is never cleared, never goes away. And over time, the virus builds up, becomes a persistent viral infection, and it may be fatal. Many of the persistent viral infections are tolerated by the patient, like cytomegalovirus, chickenpox. My mother had, what do you call that? I just talked about it too. Shingles. And she had it for like the last 3 or 4 years of her life. She had the shingles all the time. They never went away. So it was a persistent viral infection in her case. And this happens rarely. It's less than 1 in 1,000 patients. When they get the shingles never goes away. It was tolerated, and she always had the virus. The patients who have cytomegalovirus, they're constantly shedding viruses, but it's tolerated. It generally doesn't cause much disease signs or symptoms. And initially, the virus load is low, and then it climbs over time. Like I said, usually the persistent viral infections get worse over time. If it becomes tolerated, the virus will usually plateau, and you no longer get, you still have the virus being produced, but it's not produced at higher and higher numbers. It stops at a certain number, and the patient then tolerates it. Any question about any of that? Normally, when a patient gets infected, the viral infection will be an acute viral infection, like the cold and the flu. The patient gets exposed to the virus, it incubates, and then the virus number quickly replicates to a high number. This is an acute infection. The patient will be sick in this time, and then the virus quickly decreases and goes away, and then the infection ends. So that's an acute viral infection. A persistent viral infection, the patient will be exposed to the virus, and then the virus will increase over time. If it becomes tolerated, it will stop increasing and will just plateau off. If it becomes fatal, it will usually bloom in number, like that happened with HIV before the modern medication. And then you notice how the curve is broken. That's because the patient has died of the persistent viral infection. A latent infection, you will first have the acute infection, and that would be something like the child having chickenpox. And then the virus is absent. It's in hiding. It's metabolically having little or no activity, not causing any disease for perhaps a long period of time. And then the virus leaves the latent life cycle and enters the budding life cycle, and we call that a latent infection. And for the chicken pox here, this will be the shingles. Any question about any of that? All right. Any question about viruses before we leave the topic of viruses? Not hearing anything. So we'll talk about our next topic, prions. Prions are a really odd disease, or disease-causing agent, I guess. They are proteins, and they are infectious proteins that cause disease. You can have prion disease being inherited, in which case the prion disease tends to run in families. And what happens is they have a genome which is more likely to make the prion protein. And we'll talk a little bit more about that. You can also get prions by ingestion, an organ transplant, and by surgical instruments can bring the prions into a patient. Prions are proteins that cause spongiform encephalopathy, that is, vacuoles in the brain. Let me show you that. Here we have a normal brain tissue, and these are prion brains of people who have prion disease. And you can see the brain looks very unusual, and it looks like a sponge, and that's why it's called spongiform. In animals, the most common case of a prion disease was bovine spongiform encephalopathy, more commonly called mad cow disease. But there's also scrapies in sheep, and then cervid chronic wasting disease in, I guess, cervids. In humans, we don't call it mad cow disease or spongiform encephalopathy, although you could. It's usually called after the doctors who discovered it: Creutzfeldt-Jakob disease, Gerstmann, Straussler, Schenker syndrome, and I could be butchering that name. I'm not German. I don't know how to speak German either. And then fatal familial insomnia, and then Kuru. These are all human spongiform encephalopathies caused by prions. In Britain, I think it was in the 1990s, there was an epidemic in the British cows of mad cow disease. And the reason was because the British couldn't sell the cow brains. Nobody wanted to eat them. And they found that they could take the cow brains, grind them up, and then add it to grain, and then feed the grain to cattle. And so that's what they did with the cow brains. And that was a problem because one cow came down with mad cow disease, and they ground up the brains, and then the cattle ate the brains and the grain, and then the cattle came down with mad cow disease, and there was more of them, and they ground up those brains and fed them to cattle. And you can see how this would quickly cause an epidemic of mad cow disease. It also caused not an epidemic, but an infection of mad cow disease in people, which happened because people were eating tainted beef. And it also caused an infection in British cats. Around 150 British cats came down with mad cow disease. And then around 150 British citizens came down with mad cow disease. And it's believed that the people in the cats got the mad cow disease from eating tainted beef that had prions in it. Fortunately, there haven't been further cases of people who got mad cow disease from eating tainted beef in the 1990s, meaning it looks like the disease has gone away in Britain. It's curious that dogs, which were eating similar food as British cats, did not come down with mad cow disease. They must have been resistant to it, at least when they consume it in their food, because it's well known that they were eating the same type of food as British cats. Any question about any of that? All right. Prions stands for proteinaceous, PR. I, infectious. And then particle isn't part of the prion, but that's what a prion stands for, why it's called the prion. Proteinaceous infectious particle. If you get this protein, you can get prion disease. Kuru was a disease seen in the Fore tribe in Papua New Guinea. So there's Papua New Guinea. Let me blow this up. The red part is the Fore tribe, where the patient were unable to walk, had the loss of the ability to swallow or chew, drastic weight loss, and it led to death. How was it transmitted, Kuru? Well, the Fore tribe had an unusual funeral ritual where when people died, they cooked the body and then ate the dead as part of the funeral ritual. And when people started eating the brain tissue as well as other nervous tissue, because that's where the prions are in the nerve tissues, they came down with Kuru because they were eating the prions. Now, the mothers noticed that some of their children were coming down with Kuru, and then they noticed also that they themselves, the mothers as well as the fathers, were coming down with Kuru. So the mothers themselves stopped eating the flesh of their dead relatives and stopped feeding it to their children. So that when the missionaries came to Papua New Guinea, it was only the men who had Kuru. And the missionaries said, eating the dead, that's anathema. You can't do that. And they put a stomp to it, meaning they put a stop to it. They stomped it out. And that actually did the Fore tribe something good. Oftentimes the missionaries don't do good things for the people. In this case, they did. They got rid of Kuru in the Fore tribe. And simply they I don't know, they passed a law or something, and they made it illegal to eat your dead relatives. And like I said, it was a good thing because the Fore tribe no longer has Kuru. Any question about any of that? All right, I've already shown you that picture. I should probably move that one up. Prions, proteinaceous infectious particles, is a protein very similar to a protein that the nerve cell makes. The nerve cell makes a normal protein called PRP superscript C, and it's a cell surface protein involved in the, not the metabolism, but the working of the nerve cell. So the cell does need this gene and the protein in order to function as a nerve cell. What happens is the cell makes the PRP superscript C protein, which is the normal cellular gene, normal cellular protein. And it comes up against the prion protein, the PRPSC. And SC stand for the Scrapes protein, by the way. Which Scrapes is a disease in sheep that European ranchers have been living with for centuries, meaning we've known about Scrapes in Europe since close to the Middle Ages. It's a long-running disease. And they know that sheep get it, and they get it from other sheep. They didn't know exactly how it was transmitted, although it is true that I think they knew that a mother could transmit it to her young. Because sheep don't normally eat sheep, so I'm not sure how it was transmitted from sheep to sheep, but it did have some way of transmitting from sheep to sheep. And then it gave signs and symptoms similar to mad cow disease, where the sheep would go in a circle and then just lie down and die. Anyways, the Scrapes protein, the PRPSC, comes next to the PRP superscript C, the normal cellular protein. And then it changes the folding pattern of the normal gene into the Scrapes gene, so that both the PRP superscript C and the PRP superscript SC have the same amino acid sequence. So they're the same protein. They're just folded in a different folding pattern. And how the Scrapes protein replicates is it changes the normal gene into the Scrapes gene, just changing the folding pattern into the Scrapes folding pattern. The cell says, whoops, I don't have enough of the PRP superscript C gene, so it makes more. The trouble is, now there are 2 copies of the Scrapes protein, and then one of them comes up to the normal gene and converts it into the Scrapes protein. The cell says, oops, I need to make more of the normal protein. And so more of the normal protein is made, and more of it gets changed into the Scrapes protein. The Scrapes protein starts accumulating in the brain cells, forms plaques, and then it eventually kills the brain cell, leading to the Scrapes disease. Any question about any of that? So that is how a prion protein replicates, comes up to the normal protein, and then simply changes its folding pattern into the Scrapes folding pattern. Once again, the Scrapes protein is the same as the normal protein, at least the same amino acid sequence. It's just folded in a different folding pattern. Now, what did I want to say about mad cow? Oh, I wanted to mention that the families that have Scrapes disease running in the family, they have a genetic heterogeneity, meaning a gene, which is more likely for the normal protein to spontaneously change its folding pattern into this Scrapes prion folding pattern. And then that protein can replicate because it can come up to the normal protein and cause it to fold into the Scrapes protein. And that is why young people do not have the prion disease, because the normal protein has not changed into the Scrapes protein yet. But it's more likely to, and as the patient ages, it will change into the prion protein, and then the patient will get prion disease. So prion disease in the families that it runs in tends to come when the the patient is at least in middle age, if not older. And that's why the gene continues. And that's because the patients reproduce when they're young. And so the gene in the family continues. And then later in life, the patient starts having the prion disease. Any question about any of that? Any question about prions? I'll mention that what the British did to get rid of mad cow disease was they incinerated all of their cattle and then hired or bought cattle from outside the country to restock their cattle. And that was an extreme measure, but that's what was shown to be necessary to control the prion disease. And I think the British did that because they realized that people were coming down with prion disease. All right, let's talk about plant viruses and viroids. First, about plant viruses. Plant viruses do resemble animal viruses in many ways. They do cause many diseases of plants, including economically important crops like beans, corn, sugar corn, and potatoes. Because of the tough cell wall of plants, the virus usually enters a plant cell or a plant through wounds in the plant, such as with a parasite, like a nematode eating on the plant, or a fungus weakening the plant, or an insect eating on the plant. Infected plants can spread the virus in their pollen or seeds to ensure that the virus is carried to the next generation of plants. Any question about any of that? Okay, plant viruses do not cause human disease, so that's all I'm going to talk about for plant viruses. Let's talk about viroids. Viroids are an infectious agent that causes disease, and what they are is a naked RNA molecule. There is no protein coat, but if you think of them as an RNA virus, they would be similar to an RNA virus with no protein coat. And when the RNA gets inside a cell, it can then replicate and cause disease. So that's what a viroid is, a naked RNA molecule that actually causes disease. Fortunately, viroids are plant pathogens. It is true that there are something like a viroid, it is a viroid that does infect fungus, but there are no viroids known to infect animal cells. And viroids do cause disease, such as potato spindle tuber viroid. Let me get you a picture of that. So here's the normal potatoes, and there's kind of a normal potato, a little smaller. And there's a potato infected with the potato spindle tuber viroid. They're spindle shape, and then they're much smaller potatoes. Any question about any of that? All right. Once again, viroids infect plant cells. They do not infect animal cells. There's no animal disease that we know of that's caused by a viroid. That's it for chapter 13. Let's begin chapter 14. As is normal, I give you a first slide showing you the name of the topic, chapter 14, Principles of Disease and Epidemiology. I list the major goals and a rough outline. Always know the terms. If you know the terms of this chapter, you will learn 50% of the material in the chapter, meaning terms are very important for this chapter. Understand the relationship of the normal microbiome and its host. We'll talk about that. 3, understand the stages of disease development. Diseases usually follow the same stages of development. And then 4, understand what nosocomial infections are and how to control them. Some terms of this chapter: pathology, the study of disease; etiology, the study of the cause of a disease. For example, the etiological agent of anthrax is Bacillus anthracis. And then pathogenesis, the development of a disease. Some more terms: an infection is the colonization of the body by pathogens. It could also be the presence of microbes where they are not normally found. Like Staphylococcus epidermidis is normally found on my skin. If it were ever to be growing underneath the skin, like in the dermis, that would be an infection, because the Staphylococcus epidermidis isn't supposed to be growing any place except in the skin. So that would be an infection. Disease is an abnormal state in which the body is not functioning normally. Disease can be caused by a microbe. And not all diseases are caused by a microbe. For example, heart disease is usually caused by a poor diet or lack of exercise in people. It can have a genetic cause, meaning there could be a genetics to it. Rarely is heart disease caused by an infection. It used to be, before the development of antibiotics, people would get strep throat, and then it would infect their blood, and then they would have bacteria growing inside their body, and the streptococcus tended to grow, I think, in the heart valves, and that could give rise to heart disease. But that's very rare today because of the antibiotics preventing. They used to have a name for that, and I don't remember if it's not red fever. It's not typhoid fever. I don't even remember the name of that. There used to be a disease name for when strep throat, the bacteria got into your blood and then started growing in the body. And I don't even remember, scarlet fever, maybe? Something like that. You should know that few microorganisms are pathogenic. Very few of them can cause disease. Most do not. Some microbes are even beneficial to the host, such as the normal microbiota. The normal microbiota is generally beneficial to the host. Every individual animal and plant has a large community of microbes found in association with that animal and plant. We call these communities the normal microbiota, microbes that permanently or nearly permanently live with the host. and they live in symbiosis with the host. And generally, they live for about the lifetime of the host. They're only found in certain locations. They are not found internally to the body, but only externally or in the mucous membranes. Disruption of the normal microbiota is actually associated with disease. It turns out the normal microbiota establishes a balance with its host. And when that balance is disrupted, it is associated with disease and impaired immune function. Many of you have probably experienced a disruption of your normal microbiota. If you've ever taken antibiotics and you ever became constipated or you got diarrhea after taking the antibiotics, that was a disruption of your normal microbiota host balance, which led to the constipation or the diarrhea. And usually the balance reestablishes in a day or two, and then the diarrhea or the constipation go away. Anyone experience an example of something like that? Anyone want to state? Okay, I'll just mention that frequently when you take antibiotics orally, you disrupt your normal microbiota and the patient may get diarrhea or constipation. Fortunately, it's usually short term. The human microbiota is found in examples shown here externally on the body, such as the skin, the nose and throat mucous membranes, the eyes, the mouth, the gastrointestinal tract, meaning the intestines, the stomach and the throat, are also a place where the normal microbiota are found. And that's internally, but it's externally in the sense that it's a mucous membrane. The lower urinary and reproductive systems, such as the urethra in both males and females, have the normal microbiota. The vagina also has the normal microbiota. Microbes are not normally found in the blood, in the cerebrospinal fluid, the muscle, the nervous tissue, and the bone. The immune system will remove any microbes found internally, like in the blood, the cerebrospinal fluid, the muscle, assuming the immune system's working. Anyways, that's the normal microbiota, microbes that colonize the host and then live with the host generally for the lifetime of that host. Like I've got Staphylococcus epidermidis on my skin, it will be there until I die. And probably it'll be there and after I die until my flesh rots away. The transient microbiota, in contrast, may be present for days, weeks, or months, but unlike the normal microbiota, will not stay with the patient for long periods of time, like the lifespan of the host. It's transient. I once had a transient microbiota. I went and visited my aunt who was had Staphylococcus aureus infection, and she was taking antibiotics for it, so she didn't think she was contagious, but she insisted on hugging me and my mother. I took my mother to see her sister. And I normally, at that time, would be testing my skin for Staphylococcus aureus. It's just something I did in the lab. And after I visited my aunt, I happened to be testing my skin and I noticed I had Staphylococcus aureus. And I said, geez, I must have picked this up from my aunt. Fortunately, I was doing it every term. So the next 3 months when I tested my skin, I tested it and I did not see Staphylococcus aureus. And then every time I tested my skin after that, I did not find Staphylococcus aureus. So that was a transient microbiota for me. And I'm glad of that, because if you don't know, Staphylococcus aureus is a human pathogen. It can cause skin lesions, boils. It can get worse, but usually it just causes boils. And so I was fortunate that that was a transient microbiota. With humans, and we're talking about Americans, about 20% of Americans have Staphylococcus aureus growing on their skin. And it's not a transient microbiota. It has gotten established and becomes a normal microbiota. These patients are much more likely to get a skin boil than someone who does not have Staphylococcus aureus on their skin. Any question about any of that? When we're talking about the normal microbiota and its relationship with the host, you should know that humans have about 1 times 10 to the 13th human body cells. And humans have about 1 times 10 to the 14th bacterial cells living on and in them as part of their normal microbiota. So that begs to ask the question, are humans more microbe than we are human? And don't let that freak you out too much, because the answer is we do have more bacteria cells than we have human body cells, about 10 times more. Here are some normal microbiota found in the nasal epithelium, the cocci shown here. This is bacteria in the lining of the stomach, spirals. That is not bacteria, that's the stomach villi. And then we have bacteria in the large intestine. This is a cocci, not a cocci, a bacillus. These are either coccobacillus or that looks like a cocci. That there is actually food being digested, so that's not bacteria. And this here is probably a normal human cell. We found microbiota in some odd and unusual places. Fairly recently, it was discovered in 2014. Females in their bladder have a normal microbiota. Previously, it was thought that the bladder was far enough inside the body that it did not have bacteria cells as part of it growing in the normal microbiota. However, females do have bacteria in their bladder as part of their normal microbiota. I don't know if males have bacteria in their bladder as part of their normal microbiota. I've looked into this, and there is no research being done on males. And I don't know why. It might be that males do not have bacteria in their bladder simply because they have a longer urethra. And so the bacteria never get to, or usually do not get to the bladder. They can have a bladder infection, but usually they do not, simply because the urethra is longer. They discovered that there is normal microbiota in mother's milk. And I'm not talking about mother's milk once it comes out of the mother, because obviously the outside of the nipple will have microbes on it. But when they sample mother's milk inside the breast tissue, it has normal microbiota in it. And so inside the mother's breast, there is normal microbiota. And it's actually important to give that normal microbiota to the mother's baby when she's breastfeeding. Any question about any of that? The third one, I'm not sure why that was a surprise. They discovered that the mother's placenta has a normal microbiota. And this microbiota for the placenta is unique and only found in humans in the placenta. If you don't remember, the placenta is the mother and the offspring's blood supply so that the mother can get food and oxygen and nutrients to the embryo or fetus. And so it's this network of blood vessels to exchange nutrients and waste products and oxygen and carbon dioxide and everything like that. Anyways, the placenta has a unique microbiota that's only found in the placenta. And I'm not sure why this was a surprise, because it's well known that the female reproductive system is open to the outside. But the normal placenta does have a normal microbiota. Any question about any of that? All right. The normal microbiota establishes a relationship with its host. It's a symbiotic relationship where the two live together. The normal microbiota can engage in microbial antagonism, where the normal microbiota can compete with other microbes, including microbes that can cause disease, meaning the normal microbiota can compete with human pathogens. The normal microbiota can prevent the growth of a human pathogen by simply occupying the niche. For example, I've got Staphylococcus epidermidis on my skin. If a human pathogen were to fall on my skin and it were to fall on top of Staphylococcus epidermidis, the human pathogen cannot get established because the normal microbiota is already there. Occupying the niche the pathogen might try to occupy. The normal microbiota can also compete for resources. For example, if the normal microbiota were to be nearby a human pathogen that were to get on my skin, and the normal microbiota used up all the resources in that location, then the human pathogen is not going to find resources and will not grow. If it does not grow, it will not cause disease. The normal microbiota can also produce acids, making the environment less favorable for a human pathogen to get established. For example, the human skin already is slightly acidic from its secretions, but the normal microbiota can make it even more acidic, making it an environment too acidic for a human pathogen to get established. Another example of that would be the urethra has bacteria in it, which make it acidic, and that can prevent a human pathogen from getting established. The normal microbiota can also produce bacteriocins. These are proteins made that inhibit the growth of similar cells, like E. coli. If it makes bacteriocin, the bacteriocin will prevent the growth of Salmonella and Shigella, but it will not prevent the growth of other bacteria in the gut, such as, let me see if I can remember this. Enterococcus faecalis, a normal microbiota found in the gut. I've always wondered why people weren't using bacteriocins like an antibiotic. And a bacteriocin is like an antibiotic in that it keeps certain species from growing. However, it's much more specific and narrow in its action than an antibiotic. Even the most narrow antibiotics typically take a whole group of cells, like either the gram positives or the gram negatives, and then inhibit those. Bacteriocins only stop certain species or families from growing, and then other families are not stopped from growing. And I did read an article a few years back now where they were experimenting with bacteriocins to see if they could use it as an alternative to an antibiotic. Any question about any of that? All right. Probiotics are the consumption of live microbes intended to exert a beneficial effect. For example, a yogurt that has live microbes in it introduces microbes in an individual, and the individual who consumes the yogurt microbes is much less likely to get a yeast infection than a patient who does not eat those microbes in the yogurt. It used to be when patients took antibiotics, the doctors told them, you should eat yogurt to help replace the bacteria in your gut because the antibiotics will decrease the bacteria in your gut. They don't tend to do that anymore because obviously you're only replacing a few species of bacteria from the yogurt. You're not really replacing the normal microbiota, but there are other probiotics you can also take besides yogurt. All right, any question about any of this? Let's talk a little bit more about the normal microbiota. It establishes itself with the host, generally lives for the lifetime of the host or close to it, and establish symbiosis. Symbiosis is where two different organisms or populations are living together. The normal microbiota forms a symbiotic relationship which is either commensalistic or mutualistic. A commensalistic symbiotic relationship is where one organism benefits and the other is not harmed. It doesn't benefit, but it's not harmed. For example, humans have in their eyeball one nematode, which forms a commensalistic relationship with humans. The nematode does not damage the eye, does not eat the patient's cells. It must be eating the bacteria that fall into the eye. It does not do the patient any harm, but it does not do the patient any good either. So it's a commensalistic relationship because the nematode gets a home by living in the eye. There are other bacteria or other normal microbiota that form a mutualistic relationship with their host. A E. coli forms a mutualistic relationship with us. We give E. coli a home. It grows in our gut. We supply E. coli with food. E. coli helps us digest our food. So both organisms benefit. Parasitism is where one organism benefits and the other organism is harmed. A pathogen forms a parasitic symbiotic relationship with its host. The pathogen benefits because it's given a home. It can then damage the host and get nutrients from the host. Like if you kill the host cells, you release those nutrients, the bacteria can use them. So that's what a pathogen does. It forms a parasitic symbiotic relationship with the host. An opportunistic microorganism is a normal microbiota that becomes opportunistic and causes disease under certain circumstances. The normal microbiota do not normally cause disease in a healthy individual. But if the normal microbiota colonize a region of the body where they are not normally found, they can become opportunistic and cause disease. For example, E. coli is normally found in the gut, and it does not cause disease normally in the gut. We're talking about normal E. coli. So it's in all people's guts as a normal microbiota. But if E. coli were to get in the ear and then grow in the ear, it would cause an ear infection. If it were to get into the lungs, it would cause a lung infection, and we call that pneumonia. So that's where E. coli is a normal microbiota, and when it grows in places where it shouldn't, it can cause disease. Another case of the normal microbiota becoming opportunistic is with individuals with a compromised immune system. They can often become infected with their normal microbiota becoming opportunistic, such as Staphylococcus aureus is found in the nose and the throat of about 50% of Americans. Only 20% have it on their skin, but on their nose and the throat, about 50% of Americans have it. It does not usually cause disease in the nose and the throat. I'm not sure why, but it does tend to cause disease on the skin. An example of a patient with an impaired immune system getting an infection from the normal microbiota could be a cancer patient. They're taking chemotherapy that weakens their immune system, and then the normal microbiota could get established, causing infection. Like all of us have yeast growing on our skin, it's part of our normal microbiota. And if the yeast blooms, let's say the patient gets chemotherapy that weakens the immune system, and then the yeast could take off growing. The yeast is normally held in check by both the immune system and by the normal microbiota. Another example of a normal microbiota causing disease and becoming opportunistic is an AIDS patient. The AIDS patient doesn't have the immune system, the normal microbiota. If it grows where it shouldn't, there's no immune system to prevent it from growing, and then the patient gets disease. Let's change to the etiology of an infectious disease. Robert Koch showed that Bacillus anthracis causes anthrax, and this is the first time a microbe was shown to cause a human disease, or any disease, actually. He used Koch's postulates to prove that Bacillus anthracis causes anthrax. Let's go through that in detail. Trying to blow this up. There we go. So if you have a sick bunny and you isolate organisms, cells from that sick bunny, meaning bacteria, and then grow them up, purify them in tissue culture, and then if you inject a pure culture into a healthy bunny, and if that healthy bunny were to become sick, why the bunny is turned on its side because it's sick, poor bunny, then that's pretty good evidence that this organism is causing disease, assuming this disease is the same as that disease there. Come on, mouse. This disease here is the same as that disease there. Koch's postulates stated that's not quite good enough. You need to show that this disease is being caused by this organism. So you must be able to isolate this organism from this sick individual. So if the disease is caused by this organism, and then the organism is further isolated from this individual, and then all individuals that have this disease have this organism, that's evidence using Koch's postulate that this organism is causing the disease. Come on in, show. So those are the steps we use to show that a given organism causes a given disease, but there are exceptions to Koch's postulate. What do you do if the organism has unique culture requirements and cannot be grown in tissue culture? For example, we think Treponema pallidum causes the disease syphilis, but we've never been able to grow Treponema pallidum in tissue culture. So we've never been able to get it purified, grow it up in culture, and then give it to another patient to see if they get Treponema pallidum, the disease causing syphilis. And it's true also of a species that causes a human-only disease. We've never given it to humans to see if it causes disease, like HIV. We think it causes AIDS, but we've never injected HIV into a patient to see if they later get AIDS. So we don't use Koch's postulate in these cases. There are some other cases too. What do you do if multiple organisms cause the same disease signs and symptoms? In Koch's postulate, you have to have one organism that causes this disease sign and symptom. What do you do if there's multiple species that cause the same signs and symptoms? You can't use Koch's postulate. Like, for example, the human cold. There are many different viruses that cause the human cold. They actually come from 3 different families of viruses, one of them being a DNA virus and 2 being RNA viruses. So you can't use Koch's postulate because more than one organism causes the same signs and symptoms. And then what do you do if some species cause different diseases? Like salmonella can cause salmonellosis, and it can also cause typhoid fever. So these are some exceptions to using Koch's postulates. What do we do when we have a virus like HIV that we think causes AIDS, but we can't use Koch's postulate to show that this virus causes AIDS because of moral and ethical reasons? What do we do? Anyone? All right, we're about to run out of time, so I'll tell you the answer, but in the future, I'll just say you guys have to think about it. What we do is we do vast epidemiological studies, and then we follow some people, meaning we study them over time. We follow some people who are HIV negative, and because of their own activities, they become HIV positive. And then we follow these HIV positives over time and note that they tend to get AIDS. And that's why we know that HIV causes the disease AIDS. Any question about any of that? If not, I'm going to stop here. And I think I'm done with the class. Let me just check to make sure. Oh, nope, we don't end at 6:20. That's the normal time. We end at 6:50 in the summer. So let me go further. Glad I didn't stop it. I'm glad I checked. Go on. All right, some other terms to know for this chapter. A symptom is a change in the body function that is experienced by a person as a result of a disease, but it is subjective. An observer cannot observe the symptom. It cannot be measured. For example, a headache is something that only one person can experience. Nobody can measure the existence of a headache in another patient. So a headache is a symptom. A sign is a change in the body that can be measured or observed as a result of a disease. For example, a temperature of 38.5 degrees Celsius, which is, I think, around 102 degrees Fahrenheit, is something that anybody can measure. So a sign is something that another person can measure. Any question about any of that? Usually I just use signs and symptoms when I'm talking because I don't really care if it's a sign or a symptom. It is a manifestation of a disease, but a symptom is something that another person cannot measure. Another person cannot know that the patient has a headache. Only the person who has it knows they have a headache. A sign is something that anyone can measure. A syndrome is a specific group of signs and symptoms that characterize a disease. For example, AIDS has a specific sign and symptoms characteristic of AIDS. A communicable disease is a disease that spreads from one host to another. Measles, tuberculosis, and chickenpox are all communicable diseases. A contagious disease is a disease that easily spreads from one patient to another. Can anyone name a contagious disease Actually, that's pretty easy. A contagious disease, one that easily spreads from one to another. Give me a disease that is communicable but is not contagious. This is a disease that can spread, but it does not spread easily from one patient to another. And I'll give you a hint. There is one shown on this slide. Anyone going to take a guess? All right, think about that. I will warn.
Chicken pox?
Say again?
Chicken pox?
No, chickenpox can easily spread from one patient to another. In fact, in the old days, they used to have chickenpox parties because they wanted their children, when they were healthy, to be exposed to it and then get chickenpox when they were healthy because it was a minor disease and you may as well get it now when the child is healthy rather than to wait when the child is sick and then get chickenpox.
But what if you already had chicken pox?
Then you could not get chickenpox because you've already had it. You could get shingles when the virus left the latent life cycle and then re-entered the budding life cycle, but you would never get chickenpox again.
Okay.
Flu?
I'm sorry?
Flu?
The flu. The flu is an example of a contagious disease. So it is not a communicable disease that is not contagious. It does spread fairly easy from patient to patient. Nobody knows a disease that doesn't spread very easily. The best one I can think of is actually, oh gosh, what is that called?
I mean, it says tetanus on your example. With human tetanus.
Tetanus is not a communicable disease.
Oh,
I'll
you're talking about the
tetanus
top still,
later.
sorry.
I'm talking about
Oh,
a communicable disease that is not.
Oh, okay.
Okay. Leprosy is actually a very good example. A disease that is communicable. It can spread from one person to another, but it does not spread easily. You have to have long-term exposure, and then not everyone can get leprosy. I think it's only about 10 or 15% of the human population that can actually get leprosy. And you need long-term exposure, so it is communicable, but it is not contagious because it does not spread easily. Another disease that does not spread easily is tuberculosis. It is communicable, it can spread, but it does not spread easily. Contagious disease is one that spreads easily. Examples of a contagious disease are colds and flus. They spread easily. COVID-19 spreads even easier. Measles and chickenpox are contagious diseases. But tuberculosis is only communicable. It can spread, but it does not spread easily. There are also non-communicable diseases, and these are diseases that do not normally spread from one patient to another. For example, when a patient gets tetanus, that bacteria is not going to spread from that patient to another normally. I can think of an example of a non-communicable disease that can spread from patient to patient, but this is very unusual. And that is if you had a cannibal eating the tetanus patient, the cannibal can get tetanus. And so the disease could spread to the cannibal. But this is a very unusual circumstance. Normally, a non-communicable disease does not spread from one patient to another. And tetanus is an example. Another one is botulism. You can get botulism by eating inappropriately canned food, usually home canned food, but it could be store-bought and canned food too. But once you get botulism, it does not spread from you, usually. So a non-communicable disease. When we're talking about disease, it's important to talk about the occurrence of a disease. To understand the full scope of disease, we need details about its occurrence. When we say the incidence of a disease, we mean the fraction of a population that contracts a disease during a specific time. The incidence only counts the new cases of the disease. It does not count the old cases of the disease. The prevalence, on the other hand, talks about the fraction of the population having a specific disease at a given time. The prevalence does count both the new cases of disease and the old cases of disease. Usually, when we're talking about COVID-19, people are giving the prevalence numbers, the number in the population that have come down with COVID-19, and that's counting both new cases and old cases. Usually when we're talking about the flu cases in a year, we're talking about the incidence, the fraction of the population that have contracted the flu recently, so only the new cases. For example, AIDS in 2007, the incidence of patients coming down with AIDS was 56,300 cases in the United States. The prevalence of AIDS cases, though, in 2007 was 1,185,000. And that's the new cases plus the old cases. Any question about any of that? All right. Some terms we apply when we're talking about the occurrence of a disease include these here. A sporadic disease is a disease that occurs only occasionally in a population. So some people say typhoid fever in the United States is a sporadic disease. It occurs occasionally, but not all the time, and usually in low numbers. An endemic disease is a disease that is constantly present in a population. So you can say that the common cold is an endemic disease in any human population. Because as far as I know, the colds hit everywhere, usually in the wintertime. Malaria is a disease that is endemic to the tropics. So you go anywhere in the tropics, you can get malaria. An epidemic is a disease acquired by many hosts in a location at a given time. So when influenza first becomes epidemic, there's a large number of cases of the flu in one location, usually one continent, and we call it epidemic. If that epidemic then spreads to more than one continent, we call it a pandemic disease, a worldwide disease. Like COVID-19 at the moment is in a pandemic. Any question about any of that? An example of an epidemic that didn't go pandemic would be SARS. The second time that SARS hit the world, it only hit China, and so it was an epidemic in China. An epidemic, by the way, is just when the number of disease cases is significantly higher than expected. Like in the flu, normally in the wintertime, we have an endemic cases of the flu. We have an epidemic if the flu cases are much higher than normally expected. So normally in the winter, we have the flu, and that's not an epidemic, it's endemic. It has to be when it's significantly higher than normal. And we usually get the flu epidemics about every 10 years when a new flu comes along and people's immune system aren't used to it, and then we get an epidemic of the flu. We'll talk a little bit more about the flu later in the course. Am I talking about that in this course or not? I don't remember. I don't think I'm talking about it in this course. Sorry, I shouldn't have mentioned that. Any questions about any of these terms? Another way of talking about disease is talking about the severity or the duration of a disease. So when we're defining the scope of disease, it's important to talk about its severity or its duration. An acute disease is a disease where the symptoms develop rapidly, the virus or disease comes quickly, and then lasts for a short period of time. A classic case of an acute disease is the human cold. The patient becomes infected with the virus, quickly develops signs of cold, and then the cold quickly goes away. An acute disease. A chronic disease is a disease that develops slowly, persists for long periods of time. Generally, it gets worse with time. Mononucleosis is a chronic disease where the mono develops slowly with time and then persists for a long period of time. Hepatitis B causing liver cirrhosis is a disease that develops slowly, persists for long periods of time, and the patient will have a hepatitis episode where the liver gets worse. Chronic diseases. Any question about any of that? We have diseases in between acute and chronic, and they're called the subacute disease, but I'm not going to test you on that, where the symptoms are between an acute disease and a chronic disease. There's an example, subacute sclerosing panencephalitis, but I'm sure none of you have ever heard of that. And I've heard of it, but I don't remember even what it is. But like I said, I'm not going to test you on that. I'm just going to mention that there is something in between an acute disease and a chronic disease. When we're talking about disease, it's important to talk about the extent of host involvement in the disease. Infections can be classified according to the extent to which the host body is affected. You can have a local infection where the pathogen is limited to a small area of the body, like Staphylococcus aureus infecting the skin can cause a skin boil. Another local infection is an abscess, where one area of the body is infected. That's a local infection. A systemic or generalized infection is where the infection spreads throughout the body. Measles, when a patient gets the measles spreads throughout the body. It will be a systemic infection. A focal infection is a systemic infection that began as a local infection. So MRSA, that's multi-drug resistant Staphylococcus aureus, can start on the skin, will start as a boil, can invade the blood, and then be carried to the blood throughout the body. So that would be a focal infection. Any question about any of that? Bacteremia is when a clinician notices the presence of bacteria in the blood. That's different than septicemia, where we have growth of bacteria in the blood. Usually, when clinicians look at the blood, they'll only note bacteremia, meaning they do not have the tools to test if there is growth of the bacteria in the blood. Toxemia is the presence of toxins in the blood. Viremia, the presence of viruses in the blood. Any question about any of that? When we're talking about the extent of host involvement in disease, the state of the host resistance also determines the extent of infections. If the host resistance is good, the patient may be subclinical. That's the third one down here, where the patient has no noticeable signs or symptoms of an infection. We call that an inapparent infection or subclinical disease, and I've talked about this earlier, an asymptomatic disease, where the patient carries the disease-carrying organism but the patient has no signs or symptoms of disease. Most of you are young, so I will mention that COVID-19 may be asymptomatic in you, where you have no signs or symptoms that you're carrying COVID-19. And that'll be about 50% of you, assuming you're young, which most of you are. The problem with that is you're asymptomatic, and then you can give the COVID-19 to somebody who it could cause a very serious disease in. An example of a subclinical disease was some people could carry polio virus and never have signs or symptoms of polio. And then they could give polio to somebody else, and that person could get polio, meaning the signs and symptoms of polio. A primary infection is where the acute infection begins and causes the initial illness. Like when the patient first comes down with a cold, the virus will be growing in the throat and you will have a sore throat, the primary infection. The secondary infection is where an opportunistic infection happens after the primary infection. The host immune system is weakened, and this predisposes the host to getting the secondary infection, and then another organism comes in and causes the secondary infection. In children who often get colds, they can later get a secondary infection, which will cause an earache. And that's an opportunistic infection. Someone who comes down with the cold can get an infection of the throat of another organism, and that's because the cold weakened the body's immune system and may have damaged the mucous membrane, allowing the secondary infection to occur. I'm trying to think of an example of that, and I'm not just coming up with something like a bacterial infection of the throat that happened after a patient got the cold. That would be a secondary infection. Any question about this slide? All right, let me mention that a latent disease is a disease where the disease-causing organism goes latent, goes into hiding, goes away. The disease-causing organism does not cause disease. The latent organism is inactive. The patient has a period with no symptoms or signs, and the pathogen is inactive. The best example is shingles, where the child gets chickenpox. The virus then goes latent, and then usually doesn't come out until the child is in old age and causes the shingles. So that's a latent disease. Herd immunity is where many people in the population are immune to the disease, and then that spreads, keeps the disease from spreading. For example, if the population is not immune to disease, and then two people come down with the disease, everyone is susceptible to that disease, and then the majority of the people will come down with that disease at one time or another. Only a few lucky will escape the disease. If, on the other hand, the population is immune to the disease, they're yellow, and then two people that are red come down with the disease, only the people in close contact with these contagious people will come down with the disease, meaning that person there and that person there. And then this person and those people are safe because the disease has to cross to this person to get there, or to that person and to that person to get there, And these people are immune, so the disease does not spread further. And that's the purpose of giving vaccines, to immunize the population to prevent the spread of disease. That does protect the immune people who are immunized, but it also protects the non-immunized people because the disease does not spread to them. With COVID-19, it was estimated that 85% of the population has to be immune to COVID-19 before the disease will stop spreading. Actually, it's probably higher than that now because each new variant that comes out tends to spread more easily. And so my guess is now we need above 85% of the population. With measles, you need 90% of the population to be immune to getting measles, or the disease will spread. And every once in a while, people say, oh, measles isn't a problem anymore. I'm not going to bother getting my child immunized to it. And then the frequency of the children who are immunized drops below 90%. And then measles, we have an outbreak of measles that year. Any question about any of that? If not, I think I'm going to stop here, and I'll be in the lab for questions, but only for the first 15 minutes. If you don't have any questions, you don't need to show up in the lab. Oops, wrong one. Come on. Okay. All right. If there's no questions, I'm going to end this here. I'll see you on next class.